So special type of chromosomes is nothing but special type of chromosomes are those chromosomes which differs in size and structures than usual chromosome. These chromosomes shows very large size and hence called as joint chromosomes. These are found usually in the insect and certain amphibian cells. Certain amphibian cells, the joint chromosomes are two types. That is, that is the polytene chromosomes and another one is lambrous chromosomes. So I have prepared some slides. See here, special chromosomes. Special chromosome means which significantly different in structure and function from the normal chromosomes are known as special chromosomes. Special chromosomes are two types, that is lambrous chromosomes and polytene chromosomes. Polytene chromosomes. First ones, lambrous chromosomes. Lambrous chromosomes is found in darkly stained, rod-shaped bodies visible under light microscopes in a cell during metaphase stage of mitosis are referred to as chromosomes. Strasburger was the first pioneer man who discovered chromosomes in 1875 and then turns chromosomes was pioneered by Waldier in 1888. Then the lambrous chromosome is special type of chromosome in which large number of loops are projected out from the chromatin axis giving a lambrous appearance. Such chromosomes are called lambrous chromosomes. Okay. Next, they are found in oocyte nuclei of both vertebrate and invertebrates and spermatocyte nuclei of Drosophila during diplotin stage. Lambrous chromosome is a first discovered by Reckitt in 1892. Sharp eggshells. लॅम्ब्रस क्रोमोझोम्स हा 1892 मध्ये रकेट याने शार्क एगशेल मध्ये शोध लावलेला आहे. Then it occurs in oocyte of vertebrate, that is shark, amphibian, reptiles and birds, as well as in some invertebrates, that is Sagitta and Sepia. Then it found in those cells which produce a lot of DNA and their cytoplasmic and nuclear volume increases. The detailed structures have been studied in the diplotin stage of mitotic division. During diplotin stage, certain chromosomes stretch out large loops of DNA causing the chromosomes of resemble a lambrous. They are visible under the लाईट मायक्रोस्कोप. तो सहजासहजी दिसत नाही, तो फक्त आपल्याला कशामध्ये दिसू शकतो, लाईट मायक्रोस्कोप खाली दिसू शकतो. कोणता म्हणजे लॅम्ब्रस क्रोमोझोम. देन नेक्स्ट, मॉर्फोलॉजी ऑफ लॅम्ब्रस क्रोमोझोम्स. ओके, each lambrous chromosome consists of main axis having two chromatids. त्या लॅम्ब्रस क्रोमोझोमला टू मेन ॲक्सिस असतात आणि ते क्रोमॅटाइड्स. Main axis have a row of granules known as chromomeres which are held together by fine axial fibers. Lateral loops in pairs projects from the chromomeres. About 1 to 9 loops may arise from single chromomere. Their size is various. म्हणजे 1 ते 9 लूप्स सिंगल लूप्स क्रोमोमियर्स तयार होतात आणि त्याची साईज काय डिफरंट असतात. They are held together at a points of chiasmata formation. ज्यावेळेस पॉइंट निर्माण होतो त्या ठिकाणी कायस्मा फॉर्मेशन होत असतो. The loop of pairs chromomeres for mirror image structures. This stage can last several months. Okay. Here the lambrous chromosome structures. This is the maternal chromosomes and second one is the paternal chromosomes. It's a zigzag spring-like shape. This one enlarged. It's just small portions and this small portion is taken in the light microscopes. This is the chromomeres. At the center, chromomeres and two loops are developed at a center of chromomeres. That is chromatin loops. One loops and another one loops. Enlarged section of chromomeres. This is the one sections. Here small portions the enlarged, then one sections of chromosomes is a chromatin loops. Chromatin loops is a balloon shaped sphere. Balloon shaped is found in the chromomeres. That is chromatids 1 and another one is chromatid 2. Okay. Lambrous chromosome transcription. The transcriptions occurs either in the whole loops or at the part of loops. At the beginning of meiosis, when DNA replication is complete, the homologous pairs lies immediately next to each other and form characteristic structures composed of 4 chromatids. Okay. The lambrous chromosome is distinguished by special high rate of RNA transcription. या lambrous chromosomes special high rate कशाचे म्हणजे RNA transcription. Lambrous chromosomes functions. The lambrous chromosomes involved in the synthesis of RNA and proteins. Each loop is believed to produce one long operon consists of respective citrons, and each locus code of RNA and loop is supposed to synthesis at a high rate because of repetitive gene sequence. Okay. These are also report that the lambrous chromosomes help in the formation of yolk material in the eggs. हा कशासाठी yolk material formation करण्यासाठी कशामध्ये egg मध्ये help करत असतो. Next, second one is polytene chromosomes. The polytene chromosome was first discovered by E.G. Bilbiani in 1888 in squares of salivary cells of Chironomus larvae. ही जे आपण गांडूळ म्हणतो का नाही गांडुळीमध्ये हा पॉलिटिन क्रोमोझोम्स आढळून येत असतो. They also occurs in rectal epitheliums and Malpighian tubules. They are many times larger than the normal chromosomes, reacting a length of 200 micrometers and are visible even under compound microscope. The compound microscope खाली देखील आपण पाहू शकतो सहजासहजी. The enormous size of to the duplications of chromonema which does not separate. According to the estimates, the polytene chromosome have 1,000 times more than the normal somatic chromosomes. Because of these chromosomes actually consist of many strands, they called as polytene chromosomes. त्या polytene chromosome मध्ये many स्टँड असतात, असे बँड असतात, ते बँड म्हणजे पॉलिटिन क्रोमोझोम्स. Morphology of polytin chromosomes. It contains five long and one short arms, reacting from a central point called centromeres. The entire eight chromomeres found in the cells. Total 8 chromosomes found होतात आणि एक शॉर्ट आहेत आणि 5 लॉंग आहेत. हं? Out of the 6 arcs, the start act present with the fused in 4. सहापैकी फ्यूज कशामध्ये 4 वर फ्यूज होत असतात chromosomes, and the largest represent fused sex chromosomes. About 80% of the DNA is located in the bands and 50% in interbands. Chromatin is darkly stained and is more than condensed that the chromatin is interbands. Intensely stained chromosome intensely stained chromosomes segment correspond to high degree of packing and genetically inactive, that is, heterochromatins, less neatly packed segments, still less distinct and correspond to segments with the genetically active, that is euchromatin. Genetically active जो chromosomes आहे तो कोणता आहे म्हणजे euchromatins आहे. This is the polytin chromosome structures. There are 8 out of it. 1, 2, 3, 4, 5, 6, 7, 8. Here one. 8 total out of 6 are longs. 1, 2, 3, 4, 5, 6. 6 are longs and 1 is the short arms. This is the fourth chromosome and the right arms of the chromosome 2, left arm of the chromosome 2, left arm of the chromosome 3, centromeres. He at the center centromeres, right arm chromosome 3 and X chromosome. This is the S chromosome. This is the enlarged chromosomes. This is here bands, that is the dark bands and light bands. This is the inter bands. This is the dark bands and inter bands. This is the mRNA. Here present the mRNA and the chromatins here. Chromatins center cells. Next, in Drosophila, 500 bands have been found in the cell in the four chromosomes of salivary glands. Of salivary gland. Okay. Chromosome in bands are at right angles to long axis of chromosomes. Band have high DNA constant, high DNA contains and absorbs UV lights. म्हणजे ह्या बँडमध्ये डीएनए कंटेंट्स असतात आणि ते absorb करून काय करतात म्हणजे UV light, ultraviolet light will be absorbed. Functionally, stages of chromosomes. Polytin chromosome forms structures that correlate with the functional state. During the larval developments of Drosophila, a series of expansions that will be called as puff happens in the temporal stage in the polytin chromosome. Chromosome puff decompose the expanded segment that represent active chromosome again, that is region of that one being transcribed. The location and duration of the pup reflect different stages of larval development. The incorporation of radioactivity labeled RNA has been used to demonstrate that DNA, RNA synthesis, sign to gene activity, transcriptions occur in this region. Which regions? That's chromosome region. Chromosome puff. There are certain interesting structure associated with the bands in the giant chromosomes called as chromosomal puffs or Bilbiani rings. What is meant by Bilbian rings? Because Bilbian is a scientist here discovered a chromosome that will be called as Bilbiani rings. The swelling are called chromosome puffs. The puffs are associated with the metabolic activity and represent areas of active RNA synthesis. Functions of polyethylene chromosomes. Increasing the volume of cell nuclei and causing cell expansion. Metabolic advantages as a multiple copies of genes permit a high level of gene expressions. In Drosophila melanogasters, the chromosomes undergo many rounds of endoreplication. They produce large amount of gene before populations. There is a tandem duplication of various polyethylene bands located near the centromeres of the X chromosomes, which result in the bar phenotypes of kidney-shaped eyes. Bar phenotype of kidney-shaped eyes. Thank you. 19 Thank you, 19 Thank you.